Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Monday Seminar Series. Before introducing today's speakers, let me remind you to ask your questions for our speakers using the Q&A icon located on the webinar toolbar. I encourage you to ask your questions as they arise during the presentation. And for our final weekly Spring 2024 seminar, we have two speakers, Dr. Francine Grodstein and Dr. Jun Lee, who will discuss their joint research on diet patterns, omics, and aging. Dr. Grodstein earned her doctorate in epidemiology from Harvard School of Public Health, and then served on the faculty at the Harvard Medical School and the Harvard T.H. Chan School of Public Health for over 20 years. During that time, she served as a scientific director for the Nurses' Health Study, and her research has included over those years a broad range of issues related to healthy aging. In 2020, Dr. Grodstein joined the Rush Alzheimer's Disease Center as professor of internal medicine at Rush Medical College. Dr. Lee earned her MD in preventive medicine and a PhD in cardiovascular epidemiology in China, and then completed her postdoctoral training in integrated omics and precision nutrition at the Harvard School of Public Health. She is currently assistant professor of medicine at Brigham and Women's Hospital and Harvard Medical School, and her research mainly focuses on integrating multi-omics data with population studies to help identify novel risk factors and pathways underlying age-related disease, and also to understand pathways through which diet and lifestyle may affect health. Drs. Grodstein and Lee began working together at the Nurses' Health Study, and they have continued collaborating with a new focus on nutrition and genomics and brain health in the Rush Alzheimer's Disease Center cohorts. And today they will discuss their joint research. With that, let me turn over to the microphone over to Dr. Lee, who will present first.
Thank you very much for the very nice introduction. So today, Dr. Grodstein and I are gonna introduce you some of the research that we conducted together, both in the large population cohorts focused on omics, mostly measured in blood, and as well as how we extend the same methodology to analyze omics in brain samples to elucidate how we can use multi-omics to understand mechanisms under aging, as well as to facilitate personalized nutrition. So first, a little bit about the study background is that, as we all know, that chronic disease has a very high prevalence, both in the US and globally. In the US, nearly 40% of the individuals has at least one type of chronic disease that include cardiovascular disease, diabetes, Alzheimer's disease, as well as different types of cancer. So in a traditional epidemiological approach, we can potentially analyze the association between a wide variety of different risk factors in association with disease outcomes, or in an intervention study, analyze the effect of different risk factors or interventions on the disease outcomes. This will help us understand the risk factors, increasing disease risk for prevention. However, most of the situations in this traditional epidemiological design, the mechanisms underlying the association between those exposures and outcomes are really unclear, and a traditional epidemiological approach cannot help us understand that. However, with the advances in high-dimensional omics technologies, we will able to measure different molecular profiles at different biological level. For example, the genetics, the epigenome, transcriptome, proteome, metabolome, or right now even metagenome. So this progress in both technologies and the progress in advanced analytics help promote a paradigm shift in epidemiology from a traditional epidemiological approach to a more of a systems epidemiological approach. So what is systems epidemiology? It refers to the approach that we integrate multi-omics technologies with the concept of system biology based on population-based epidemiological studies to help develop, to help understand the mechanisms underlying disease and mechanisms through which different risk factors affect disease, which could eventually advance our understanding of both disease and better preventive strategies. So let's put that context into the nutrition epidemiological studies. So in a traditional way, we can measure nutrient intake through dietary questionnaires or do randomized trial of different food groups or dietary patterns. And then we follow up individuals and analyze the relationship between the nutrient dietary intake with different health outcomes. However, as we also know that our human body interact with nutrient and foods in a complex manner to digest, to metabolize, absorb, and utilize those nutrients. Therefore, a variety of factors of human body could affect how we respond to the diet we eat. For example, the individual metabolic capacity and individual genetic constitution, or even how different and similar our gut microbiomes are. So all those factors could potentially affect how we respond to diet. So therefore, if we in the nutritional study, if we incorporate the analysis of genetics, gut microbiome, and metabolomics, those omic profiles can potentially help us understand what at the biological level, what is changed after we eat those foods and how that biological changes may affect disease risk. So it can help us understand the mechanisms underlying the association between diet and health. And on the other hand, if, for example, if we identify molecular signatures in blood or even gut microbiome using fecal samples, it can help us understand or build biomarker tools for us to monitor the metabolic response of each individual to food intake, which can facilitate the precision nutrition and a better prevention. So how that concept may help us with disease prevention. So there's a precision prevention model. In the regular, traditional approach, say we have a population, different colors here representing people with different characteristics. In a traditional approach, we can apply the same preventive strategies to the whole population. However, the strategy may be effective on the population level, but at individual level, some individual may have different responses to the preventive strategies, and some may receive a much higher benefit, but some may not. But however, if we can understand the intrinsic factors and extrinsic factors such as preference, environment, psychosocial factors, how those factors affect our adherence and biological responses to different preventive strategies, we can potentially stratify all of the individuals into different strata and then assign different or more fitting preventive strategies to individuals within each strata. Therefore, we can maximize the potential benefit of the preventives to the whole population and each individual. And in this talk, we're gonna split the talk into two parts. So the first part, I'm gonna introduce some of the study we did with blood metabolomics integrated with others omics to understand metabolic mechanisms potentially underlying the association between diet and health. And then Dr. Grodstein will go further ahead to explain some of the new exciting studies we did with brain omics. So first, about the blood metabolomics and omics studies, a quick review of the study population we use before we go into the results. Most of the analysis we did are focused on the three Harvard cohorts, including the Nurses House Study, the Nurses House Study 2, and Health Professional Follow-up Study. Those three are prospective studies enrolled many decades ago. In total include over 200,000 individuals. All of the participants were followed up every 2 years for their lifestyle factors, to collect their data on lifestyle factors and health and disease outcomes. And we also collected data on diet every 4 years using a validated food frequency questionnaire. And we had blood sample collected at different time points. With the baseline blood collection, we have over 14,000 individuals measured metabolomics at a very early time in the during the cohorts. Another study we have used frequently in our metabolomic study is a PREDIMED study, which is a dietary intervention trial conducted in Spain among individuals at high risk for cardiovascular disease. A total of over 7,000 of individuals were enrolled in the trial. The trial is to test the effect of 2 different Mediterranean diet with additional supplements of either extra virgin olive oil or nuts over a controlled healthy diet for the primary prevention of cardiovascular disease. So in this trial, we also have a large number of sample individuals. We measured blood metabolomics at both baseline and year 1. We also have some of our study. We also have replication conducted in other cohorts. Here we show example for Hispanic community health study and study of Latinos, which is a community-based cohort focusing on the health of U.S. Hispanics and Latinos. The whole population include over 16,000 individuals, and for metabolomic study, we have over 7,000 of individuals. And we also have a Women's Health Initiative, the WHI, which is a nationwide study incorporating both intervention trials and observational study for the health of postmenopausal women. And we have metabolomics data for over 2,000 of individuals in this cohort. So then we move to 2 studies. The first study we want to is to focus on how we can use metabolomics to measure both dietary adherence and metabolic response. And why is that? As we previously mentioned, that after food intake, there are complex biological processes going on within our human body to help digest and metabolize food. And all those processes can be affected by a lot of different factors. However, with plasma metabolome, the metabolites are intermediates or products from the biological processes. Therefore, the metabolome, which measures all of the metabolites we can detect in the plasma, may have the capacity to reflect metabolic homeostasis as a result of the combined effect from both diet and other factors. So under this, we want to ask the question whether a dietary pattern, whether we can identify a specific blood metabolomic profile to represent both the intake and the metabolic responses to a specific dietary pattern. To answer this question, we first use a Mediterranean diet as an example to study this question. And this is because the Mediterranean diet has been shown by both prospective cohort studies, by many prospective cohort studies, as well as many large dietary intervention trials to reduce CVD risk and the effect size has been proving very robust across different populations. So in the first study, we use a Mediterranean diet as an example to examining whether we can identify a plasma metabolomic profile for the Mediterranean diet. So for this study, we used a two-step approach. First, we conducted the analysis in the pre-med trial. As I mentioned previously, this is a dietary intervention trial conducted in Spain to test for the primary prevention of cardiovascular disease by the Mediterranean diet. And then we move forward to conduct replication studies in the nurses' health study to see whether the same metabolic profile in response to the Mediterranean diet can be replicated in an entirely different population, the US cohorts. The first question of the study is that we want to develop, so in this cohorts, we measured blood plasma metabolites using the LC-Mass platform at the Broad Institute, and after quality control, 302 metabolites were qualified for analysis. So the first question is, among all of the metabolites we can detect with good quality in the plasma, can we identify a specific metabolic signature assessing the metabolic response and adherence to the Mediterranean diet? So for this analysis, we use a machine learning model we call ELASTNet regression to regress the Mediterranean diet adherence on all of the plasma metabolites. And with this model, we identified a composite of 67 metabolites that can characterize the metabolic states related to the Mediterranean diet adherence. Of the 67 metabolites, we can see their composition in here. We have an enriched set of amino acids and also other types of metabolites, including different lipids, carnitines, and cofactors and vitamins. And with this metabolic signature, with these metabolites, we calculate the metabolic signature in both pre-med study and the nurse health study, and we and HPFS, and we found that there is the metabolic signature showed a robust correlation with the Mediterranean dietary score in both study population, which essentially showed that the metabolic signature can potentially capture some variation in the dietary adherence. And then we see that among individuals with the same Mediterranean dietary score, we do see that there's a huge variance between person variance in the metabolic signature across people with the same Mediterranean dietary score. So we want to understand what kind of what could potentially be the sources of such between person variation. One thought is that even though people reporting the same Mediterranean dietary score, their actual food choices across all the food emphasized by the Mediterranean diet could be different. So after some epidemiological analysis, we found that the differences of food choice can explain around 4 to 15% of the variation in the metabolic signature. And there's another source of potential variation to this is potentially because of genetics. As we know, each person may have different genetic variants, especially on particular metabolic pathways, and those genetic variants may have an impact on dietary metabolism. So we did a GWAS analysis, genome-wide association analysis on this metabolic signature we identified. And we found that genetics in total can explain about 12% of such a variation in the metabolic signature. And actually, interesting thing is, with the GWAS, we identified about 6 different loci associated with this metabolic signature. Among those 6 loci, there are several loci located on genes related to fatty acids or amino acid metabolism. One interesting example is this gene cluster, the FADS1 to FADS3 gene cluster. This gene cluster decodes the fatty acid desaturate, which is essential in fatty acid metabolism. And with some with other additional analysis, we also find that the top hits, the top genetic variants we identified in here is also associated with gene expression of the fatty acid desaturates in multiple tissues related to cardiovascular disease risk, including adipose tissue, coronary artery, and some of the heart tissue. So this evidence shows that the metabolic signature could also potentially capture some of the between individual variability in dietary metabolism that is caused by genetics. So the final question to this study is that we identify this metabolic signature that can measure adherence to the Mediterranean diet and is also affected by genetic variants, and whether this metabolic signature of Mediterranean diet is also associated with CVD risk as what we discovered for Mediterranean diet itself. So we conducted prospective cohort analysis in both pre-med study and the 3 Harvard cohorts. What we found is that with the Mediterranean dietary score, it has an inverse association with cardiovascular disease risk. However, this inverse association attenuated to non-significant after further adjusting for the metabolic signature. However, there's an inverse association between the metabolic signature and CVD risk that is consistent before and after the adjustment for the Mediterranean dietary score. Similar association pattern is also observed in the 3 Harvard cohorts with the metabolic signature showing consistent inverse association with CVD risk. And with the GWAS analysis conducted, we can also do Mendelian randomization analysis to see whether genetically inferred metabolic signature is also associated with cardiovascular disease risk. Consistently, we do see a significant inverse association of the genetically inferred metabolic signature with both coronary artery disease, stroke, as well as ischemic stroke. So this study is actually based on reproducible findings in both Spain and 3 Harvard cohorts. We identified this metabolic signature that measures adherence and metabolic response to the Mediterranean diet and predicts future CVD risk. So with this study conducted, an interesting next question is, in the epidemiological studies, we have already identified multiple different dietary patterns that show robust association with cardiometabolic disease and other aging-related disease. Do these different types of diets lead to similar or different metabolic alterations? So in an ongoing study as extent of the previous study I just introduced, we examined 8 different dietary patterns, including 3 guideline-based diets, such as healthy eating index pattern, alternate Mediterranean diet, and DASH diet, as well as 3 different plant-based diet and 2 magnesium-based diet, including the pro-inflammatory diet and dietary index for hyperinsulinemia. And the idea is that we want to see whether the 8 different dietary patterns is associated with similar or different metabolic responses as represented by their metabolic signature, whether that metabolic signature is associated with subsequent diabetes risk, and how gut microbiome and genetics may play a role in affecting the metabolic signature. So a quick overview of the results. We did identify 8 different metabolic signatures, each consisting of a variety of metabolites across different biological functions, strongly correlated with the 8 different dietary patterns in a variety of different study populations. And also, we did see that all of the metabolic signatures for these 8 different dietary pattern indices have a strong association with subsequent risk of type 2 diabetes that is consistent both before and after adjusting for their corresponding dietary pattern indices. So here comes to the interesting question, as we mentioned, that different diet dietary pattern themselves has similar food component and distinct component. And we're interested in how these different dietary patterns may lead to different metabolic response, and whether similar or different metabolic response may drive or mediate different differently in terms of disease association with disease. So then that's why we further examined the overlapping and distinct metabolites across different dietary signatures, and how the association between the association between diet and T2D risk was mediated by different parts of the metabolites. So here is an example for the 3 guideline-based diet. We observed that for their metabolic signature, there's a large number of shared metabolites than distinct unique metabolites. And for example, for Mediterranean diet, the overall association between Mediterranean diet and type 2 diabetes risk was 38% of the association was mediated by the overall metabolic signature for the Mediterranean diet. However, a large portion of those association was mediated by the overlapping metabolites across all 3 dietary pattern signatures, whereas unique metabolites associated with Mediterranean diet only mediate very little of the association. Similar observation was found for AHEI and DASH as well. This observation was not the same for the other dietary patterns. For example, we did exactly same analysis for the plant-based diet and between mechanistic-based diet. A specific example is for the magnesium-based diet. Between the pro-inflammatory diet and the hyperinsulinic diet, we found that their overlapping metabolites is relatively limited, and there's a large number of unique metabolites for each of the dietary pattern. And for the overall association, some of the association was mediated by overlapping metabolites, but there's also a very large or significant portion of the association between each dietary pattern that is uniquely mediated by metabolites unique to that dietary pattern. So this essentially shows that for guideline-based diet, the majority of the association between diet and type 2 diabetes may be led by the shared dietary component and their metabolic effects. However, for other dietary patterns, both the shared dietary components and the unique dietary components may drive specific metabolic effects that will lead to different association with diabetes risk downstream. And a quick review that with all these metabolic signatures, we also identified, similar with the first study, that genetic variants regulating different associated with metabolic signatures of different diets. Many of them are related to fat metabolism, ceramide metabolism, and amino acids and bile acids. And we also find gut microbiome profiles that associated with the metabolic signature. An interesting example is for the pro-inflammatory diet. Among people with specific gut microbiome compositions, even though, so among people eating the similar level of pro-inflammatory diet, we found that if person carry different gut microbiome compositions, their metabolic response could be significantly different. So that essentially shows gut microbiome may also affect how each person responds differently to the same dietary pattern. So in general, these extension studies based on reproducible findings from ethnic diverse cohorts, we identified and replicated blood metabolomic signature that measure the adherence and metabolic response to various dietary patterns and can also predict the type 2 diabetes risk. A final piece of that analysis, also a follow-up analysis, is that we also find that the metabolic signatures to those 8 different dietary patterns also have associations with all-cause mortality, but more significantly to healthy aging that is defined not only by how long you can live, but whether you can live healthily without major chronic disease and being physical, cognitive, and mentally intact.
All right, switch here. Okay, we're going to switch up on you. If hopefully you've had a chance to digest all the really interesting data June showed you. So what we're going to show you in our remaining time is how June and I have worked together to take this model of systems epidemiology and apply it to dementia research. So as I'm sure most people know by now, dementia has really become one of the foremost problems in medicine and public health, with tremendous numbers of people living to older ages, developing dementia, but real challenges in understanding mechanisms underlying the development of dementia, because, unlike for cardiovascular diseases that June was just showing you, with dementia, the target organ is the brain, and it is obviously very hard to access the brain to understand what may be leading to dementia. So we, took this model of using the interplay of diet and omics to understand possible mechanisms in dementia that could lead us to discovering new mechanistic pathways and then potentially new interventions. Go ahead, next slide, June. So to do this work, we are focusing our research in the cohorts at the Rush Alzheimer's Disease Center. These cohorts are extremely unique. They are longitudinal community-based cohorts that do annual clinical and cognitive evaluations. The unique part of these cohorts, though, is that the participants, when they die, have already agreed to donate their brains to the Alzheimer's Disease Center so that we can actually look and see what is happening in the brain and with brain tissue. So again, we have the combination of clinical data during life and then brain tissue after death, allowing us to begin to understand what mechanisms might be. So just to step back a minute, I know June was talking all about Mediterranean diet and other diets. So just here we are examining the MIND diet. The MIND diet was actually developed by Martha Claire Mars at the Rush University Medical Center. And it's kind of a hybrid of the Mediterranean and DASH diets, with the goal of tailoring it a bit to focus on foods and nutrients that have been found to be related to dementia. So the MIND diet focuses on higher intakes of whole grains, green leafy vegetables, nuts, beans, berries, fish, poultry, olive oil, and wine, with lowered intakes of red meat, butter, margarine, pastries and sweets, fried and fast foods, and cheese. And, similar to the other diets, it's developed into a score to measure adherence. So just as a quick reminder, these were some of the initial studies done by Martha Claire Mars. The left side is cognitive decline and the right side is dementia risk, which very clearly show that higher adherence to the MIND diet is strongly related both to slower rates of cognitive decline as well as lower risk of dementia. So this is just to map out how we took, the systems epidemiologic model and applied it here. So again, we start out with the ROS-map cohorts. At the time that these analyses were done, there were about 1,200 brains that we had access to, and they all had bulk gene expression, RNA-seq, available in the dorsolateral prefrontal cortex. In addition, a set of those participants had not only donated their brains, but during life had completed food frequency questionnaires. So 1,200 people had gene expression available in their brain tissue, and a subset of about 500 had dietary data available. So, just as June did with the blood specimens, we used elastic-neck regression and identified a transcriptomic profile of the MIND diet. I'll just add here quickly that there's a lot of interest, of course, in metabolites in the brain. But when we first started this research, we started out looking at transcriptomics only because the largest set of specimens at Rush had already had transcriptomics measured. So this was just a nice entryway into our experiment to see if the systems epidemiology approach could indeed translate to the brain. So we aim to develop a transcriptomic signature of the MIND diet, and then again, take that transcriptomic signature and see if it was related to cognitive decline and dementia. And so this was the first step of our results. So we indeed were able to, about the 17,000 genes, which are measured in the RNA-seq, we were able to identify a profile of 50 of these genes that together were most strongly related to MIND diet adherence. 23 of those genes were positively related to the MIND diet, meaning that higher levels of their expression related to higher adherence to the MIND diet, and 27 of those genes were, inversely related with the MIND diet. All right, June? So in our next step, we then took a separate replication cohort. So that is the middle figure in this slide, where you can see we took an independent data set of 700 people. We then calculated their transcriptomic profile of the MIND diet and looked at it in relationship to slopes of cognitive decline. And you can see here that we did find that higher score on the transcriptomic profile was strongly related to better slopes of cognitive decline, meaning cognitive decline occurred at a slower rate in those who had higher transcriptomic profile score. And then we similarly, in those same set of people, also tested to be sure that the transcriptomic profile score was related to dementia as well. And that's the bottom two bars. You can see that, in our independent set, the transcriptomic profile score also was strongly related to lower risk of both MCI and dementia. So the higher your transcriptomic score, the lower your risk of MCI or dementia. And again, we saw that both in the strict replication cohort, as well as when we combined the discovery and the replication cohort. So, really incredibly exciting evidence that we can use the MIND diet to understand, genomic level regulation of cognitive decline and risk of dementia. So we did do, a little bit of extra work, really taking advantage of the wealth of existing data in the Rush cohorts. We also have a subset of participants in whom we've actually done single cell transcriptomics. So we can really understand, we have data on 7 different brain cell types. So we took out some of the individual genes from our transcriptomic score, and we then looked at expression of those genes, again, in 7 different single cell types from brain. And And the top set of results are for relationships with slopes of cognitive decline. The bottom sets are relationships with odds of dementia. And what we see here is that across these two different, complementary outcomes, there were two genes which particularly stood out in terms of, consistent relationships for cognitive decline and for dementia, and also consistent relationships in specific cell types. The first one that really stood out was IGSF5, which again was related to expression of IGSF5 was related to cognitive decline and to dementia, even in very specific cell types, here specifically astrocytes and neurons. And in addition, this particular gene has been linked to cerebral blood flow, hypertension, as well as worse memory, suggesting that potentially the mind diet may act through vascular mechanisms. The other gene that we found, really consistently was TCIM, which is a gene that's crucial to neuron survival and synaptic plasticity. So potentially two really interesting pathways by which the mind diet could eventually influence the risk of dementia. And again, two pathways that could be explored in terms of trying to find new ways to prevent cognitive decline and dementia. So we also did some mediation analyses to understand how, to what extent some of these interesting genes we found might mediate the relationship between mind diet and dementia. So these analyses were done in the subset of participants who had filled out a food frequency questionnaire so we could assess their mind diet, and then also had transcriptomics available. And when we looked at genes which may be mediating the association between the mind diet and cognitive decline, we did identify 3 specific genes whose expression appeared to mediate, up to 30% of the association between mind diet and cognitive decline. And you will notice that one of them is that TCIM gene that we had identified before as well, even in specific cell types. We did the same thing then with the mind diet and dementia. And, again, we're able to identify 5 specific genes that mediate, quite a large proportion of the association between diet and dementia. And here again, you'll see that we do pull out the 2 genes we mentioned in the previous slide, TCIM again, and IGSF5. So, a lot of evidence through many different types of analyses of, potentially 2 specific genes of interest, which, may drive risk of dementia as well as slopes of cognitive decline. So just to summarize, we were able to identify a brain transcriptomic profile that correlates with mind diet and is strongly associated with rates of cognitive decline as well as lower risk of dementia. So, just a beginning of a way to use everything we know about diet to identify novel mechanisms for cognitive decline and dementia that could eventually lead to new therapeutics. Obviously there are, limitations here, especially because the only way we can access the brain is postmortem. So, directionality can be hard to understand. Did, maybe dementia lead to altered gene expression, or does altered gene expression lead to dementia? And although I'm not going to take the time to show it here, June also did do the Mendelian randomization analyses for this work And we did indeed find a suggestion that directionality is indeed from the transcriptomics to dementia, as we didn't see Mendelian randomization analyses didn't show the reverse direction. So, just one small piece of this, but, we do have evidence that it is indeed transcriptomics leading to dementia and not the reverse. We're starting to run out of time. So I'm going to we were going to kind of show you we have started now doing some metabolomics research in the brain tissue, but we'll save that for another time. You can look out for our eventual publication on this. And we'll just, we're going to skip quickly to some of our future, just to map out future research. Based on the preliminary work we did with the transcriptomic scores, June and I now we have a new R01 where we're going to measure metabolomics in all of these 1,200 brain specimens, and start to look at brain metabolomic signatures for healthy diet and how they relate to cognitive decline, dementia, as well as cognitive resilience, as well as eventually incorporating, sort of multi-omics to really understand pathways leading to dementia. And we did want to quickly show, the whole, host of people who contribute to all of this research, both the initial work that June showed you on blood biomarkers, and then on the next slide, the tremendous team at the Rush Alzheimer's Disease Center that makes those cohorts possible. And we do like to always end on a slide to remind everyone all of the data at the Rush Alzheimer's Disease Center are very easy to share. I know many people in this group already use the Rush data, but please, go to the website here. You can see all the data available, as well as the very simple processes for sharing the data. June and I were able to come together and do totally new things with all these data. And obviously, the more people who are working on it, better.
Thank you both for a really fascinating presentation. And we do have questions in the queue already. And the first question comes from Daniel Promislow, and he says, very exciting work. Regarding the GWAS for the diet metabolome interaction, can you clarify if the GWAS signature is for difference in adherence to the diet or in how the diet impacts the metabolome? That is, a gene by diet interaction or on the metabolite level. If it's about adherence, do you also see an effect of the effect of diet on metabolites? And can you characterize the specific genes involved? It's a long question. All right.
Yeah. That is a very interesting question. So I didn't say this very specifically, but for the GWAS of the metabolic signature, we actually did 3 different models. The first one is to do the GWAS for the dietary index score itself, which essentially measure dietary intake. So GWAS of dietary intake. And then we did the second model of the GWAS for the metabolic signature. And then the third model is to test for the interaction between diet and genetic variants in association with a metabolic signature. So we did 3 different models. For the model of for the GWAS of diet itself, we actually, across 8 different dietary patterns, we didn't see very strong associations, mainly that from the whole GWAS, we didn't see very meaningful top hits across all 8 dietary patterns. And in the second, the slides I show with a lot of top hits are actually from the GWAS directly testing the genetic variance effect on the metabolic signatures. And in the third model, where we tested for the gene-diet interaction effect on the metabolic signature, we did not see a lot of significant interaction effect between diet and genetic variants in association with metabolic signature. We only see 1 or 2 signal for pro-inflammatory diet, but not for other dietary patterns. So the majority of effects still on of the genetic variants is still on the metabolic signature themselves.
Thank you. Second question comes from Zhan Dang Wang, and he says it is very interesting that you're able to identify a metabolic signature that measures metabolic response to the Mediterranean diet and predicts CVD risk. I was wondering first if you could examine the signature in associations with cancer development, and second, if you could find any inflammatory signature which could be a mechanism linking the dietary pattern in chronic diseases such as CVD, diabetes, dementia, or cancer.
Both questions are very good questions. So for the first question about cancer development, we actually have our collaborators in the same cohort starting to look at the association between our signatures with different cancer types. So we have data on breast cancer and colorectal cancer. That studies are already ongoing. So I hope hopefully in the recent near future, we can see some of the results showing this signatures association with different cancer risk. In terms of inflammatory signature, actually, this is a wonderful idea for potentially next step research. As we know, there after metabolomics, the technologies on proteomics also developed pretty fast. And some of the proteomics technologies can measure protein profiles focusing on a variety of different inflammatory proteins, biomarker cytokines. So we could potentially apply the similar approach to proteomics to identify proteomic signatures for diet and mapping out the pathways those protein involved in, including inflammation and other immunological or metabolic hormonal pathways as well.
Okay, next question comes from Sarah Booth, who says, nice talks, and then asks, do you use any dietary assessment methods that do not rely on memory?
Yeah, actually, that is a very, very, very good question in terms of nutrition epidemiology. The most feasible way to assess dietary intake in large sample size and able to do that repeatedly is based on full frequency questionnaire or 24-hour dietary recall. So that was memory based, and so the majority of the data are memory based. We do have some of the 2 different types of data that not mainly based on memory assessed diet. One is we have sub-studies within these cohorts having data on 7-day dietary record, which people write down everything they eat at the time they eat for 7 whole days. And in the second study that I introduced, the primary development of the metabolic signature was based on the 7-day dietary record, which is have less memory recall bias for that data. Another type of data we use for dietary intake that has minimum impact on by memory is we do have several dietary randomized trial of dietary intake. For example, the PrettyMed study, we have their intervention arms and versus control arms. We have other dietary trials such as the Central trial and Direct Plus trial, which are weight loss trials that we gave participants specific diet during the whole intervention period. So that was another example. But in general,
sorry, Juna, I'm going to add one more thing here for Sarah.
Yeah, sure.
I mean, we did not sort of focus on this piece perhaps enough, but the analyses we showed you, I think the concern you expressed is probably bigger in the Alzheimer's disease center cohorts, which are, much older people than in the cohorts that June was showing. But we did, so all the analyses that we showed using diet, brain transcriptomics and dementia, we did expressly use, only their first administration of the food frequency questionnaire, and we excluded people who had dementia. So those analyses, are not subject to bias due to people with dementia having problems answering the food frequency questionnaire. So although we can't use different methods of dietary assessment to minimize that problem, we can use, sort of epidemiologic methods of, excluding certain people to be sure that it's not that dementia is leading to the diet as opposed to diet determining dementia.
Yeah, that's totally true. Thank you, Fran, for adding that. But essentially, in observational study, the dietary assessment has been easier, and there's several efforts at the national level pushing towards for better biomarkers to assess diet together with the questionnaire, of course.
So we've come to the top of the hour. We have one last question that I'll present, and that's from Yang Hu, who says, "Thanks for this interesting talk. In the data in the RADIC, are the metabolic data, cognitive data, and diet data all longitudinal?"
So the diet data are longitudinal and the cognitive data are longitudinal. All of the, data from the brain tissue is obviously postmortem. So it's not possible to measure brain levels of gene expression or of metabolomics other than after people have deceased. So yeah, I mean, that is one of the challenges, but at this, and that's why I mentioned, we do the Mendelian randomization analyses to try to kind of determine the longitudinal nature of this. But, ultimately dementia is just a much harder puzzle to solve because of the fact that, you really can't measure anything in brain tissue until after people have deceased. So, it's one of the many reasons that this is a much more challenging disease than so many other of our chronic diseases.
Well, thank you. I want to thank you both for a very interesting presentation.
Thank you.
I look forward
Thank
to
you.
seeing the future work coming out of your collaboration.
Thank you.